Okay, welcome back, everybody. Okay, so before we get rolling, great job on the exam on Monday. For the most part, those looked excellent. Our median and our mean were both somewhere in the B range. Median was an 85. For those of you who are getting a little bit of a slow start to this, reminder, you're always welcome to come go over your exam with me during office hours. In general, I have time blocked out on Monday mornings, Tuesday mornings, and Tuesday afternoons. I think there's a link, but if the times on my schedule don't work for you, we can shake around my schedule and find another time to do that. A second reminder for those of you who are in this section last time, and potentially new information for some of the rest of you, we do allow one retake of your choice at the end of term. So if you're not happy with your score, keep studying this stuff, know it by May. You can take a second stab at it then during finals period. Okay. Questions at this point? Okay, cool. So we will get moving and finish up talking about the blood. We're going to be specifically focusing on clotting. So we're going to tap back at the little bit of hemostasis that we began last time. Okay, so when we were talking last week before the exam, we had our little teaser about how we keep our blood in our body when we get injuries, when we get cuts. So big picture process. The first thing that happens is when you get a cut, you get a vascular spasm. That's just an intrinsic response of the blood vessel clamping down. And then when we actually start to go towards blood clot, we're going to need to think about these special sort of fragments of cells called platelets. They're going to need to form a platelet plug. And that's the precursor to the formation of a full regular old blood clot. Okay. So we're starting with damaging the blood vessel. We had our vascular spasm happen. We're going to pick up with the platelet stuff here. Okay. So our platelets are little fragments of cells. So they do contain some cellular machinery, but no nucleus. They do need those organelles and granules in them because they're going to secrete stuff. So they're going to be secreting hormones. They're going to be secreting chemicals that add to how they behave in your body. Okay. So the platelet's job is to form a little bit of a plug at the site where a vessel has been damaged. Now, this isn't enough to totally stop blood flow out of a cut. This is not the end of the process, but it's necessary in order to kick off the process of blood clot formation. So you need platelets for this. If you have a platelet disorder, getting a cut becomes a lot more dangerous because you are unable to form this platelet plug or unable to form it efficiently. And in that case, you are going to be in real danger, even with minor injuries, if you're unable to kind of staunch the flow of blood in those situations. Okay. So we had ended kind of with this workflow of what's going on with our platelet plug last time. Okay. Okay, cool. Okay, so we're thinking about our blood vessel, right? So our blood vessel, let's think arterial, capillary, whatever, it doesn't matter at this point. Okay, so we got a blood vessel and we're thinking about an injury, right? So we're going to be thinking about a gap. Let's make it scissors since me drawing a knife is not working out so well. Okay, so what happens here when you get a cut is you're exposing layers of that blood vessel. So we talked about the different layers of blood vessels progressively as we talked about, okay, what's in the wall of an artery, what's in the wall of an arterial, et cetera. But we know that for sure our blood vessels have an endothelial layer. Right, so the endothelial layer is that lining of the blood vessel. But also, in addition to this endothelial lining, we've got other stuff around here. We've got connective tissue, maybe we've got smooth muscle, right? So we have other stuff surround it. Better? Okay, stopped. Okay, so when we're talking about exposure of the subendothelium, what we're talking about now is that you are some molecule swimming down through your bloodstream, right? Usually you're just touching these endothelial cells, but when we have a cut, we now have contact between the contents of the blood and the subendothelium, so like the other layers of the blood vessel. Okay. One of the particularly important things that we have in that connective tissue around a blood vessel are collagen fibers. So that's kind of a structural piece of that tissue surrounding our blood vessel. And when we have some certain contents of the blood, including this particular clotting factor called von Willebrand factor, when it comes in contact with collagen fibers, it starts to behave differently. So it kind of triggers it. Okay. So when we have the von Willebrand factor coming into contact with the collagen fibers that have been exposed, this is going to kick off the binding of platelets. So the platelets are first going to adhere. So adhere just means like stick to. They're going to stick to that area where we had the exposure of the collagen. They are going to start to become sticky as they secrete out a variety of different chemicals and signaling molecules. And that's going to cause them to aggregate into a bigger lump, which is how we're forming the platelet plug. I'm trying to figure out what this is. Okay, I'll just hold it like this. That'll be great. Okay, so as our platelets collect, they're aggregating, they're becoming a larger lump, a larger collection, and they're releasing these signaling molecules. So they're secretory products. One of the things that they're secreting is something called ADP. A particular thing that ADP does is increase the stickiness of the platelets. So when we say the platelets have adhered and become sticky, part of that is because of ADP. So ADP actually also has a little positive feedback loop, increasing how much ADP is produced. So as soon as we start to have these platelets adhering and aggregating and producing the ADP, we get more and more and more platelets kind of lumping together. Now, there are other things that those platelets are secreting. So they are secreting both serotonin and epinephrine, which at the site of damage is going to cause some vasoconstriction. So this is a little separate from the vasospasm. So the vasospasm was just an intrinsic response of the blood vessel as a whole preceding all of this. And now we're causing a little more vasoconstriction when we release serotonin and epinephrine. Finally, we're going to have a couple more chemicals that are being released by the platelets that are going to facilitate the formation of a stable blood clot a little further down the line. There's also an additional kind of signaling molecule involved in positive feedback loops with this platelet aggregation called thromboxane A2. And we'll take a look at that when we look at the image of what's happening with the platelets. Okay. Before we look at the picture as a whole, I do also want to note that we don't always want to be forming platelet plugs, right? So these processes are happening because of things that are in your bloodstream kind of all the time, right? So we don't want to trigger this platelet plug formation unless we have a cut. And when we have a cut, we only want to trigger it right where we have that exposure of the subendothelium and the collagen. We don't want our platelet plug as it grows and aggregates spreading out to the side to healthy tissue, because then when we're going to be in danger of kind of blocking the whole blood vessel. So in healthy endothelial tissue, so the tissue next to your cut that hasn't been damaged, we are releasing some chemicals that like suppress this process. So prostacyclin is one of those chemicals that these healthy endothelial cells are producing, and nitric oxide is the other one that we're interested in. So these are both going to suppress the formation of the platelet plug and restrict its size. Okay, let's take a look. All right. Let's pick blue. Okay. So here we see that site of injury on our left-hand side. Okay, so we see the site of injury here, and we see that collagen exposed, right? So we can think of our first step of this platelet plug formation as that platelet adhesion. So we would have had our von Willebrand factor binding to the collagen, starting this off. Our platelets adhere a little bit here, and then they're aggregating, right? So they're collecting. So we see this bigger group of platelets here. And here we see some of the secretory products. So we don't have the serotonin and the epinephrine on here that would be constricting the blood vessel, but we do see the ADP, and this is our thromboxane A2, right? So we can see that positive feedback loop with ADP, making more and more platelets stick, making this stickier and stickier and stickier. And we can see how thromboxane A2 is also involved in that process. So you see that the platelets are producing it, right? And it's kind of amplifying that ADP stickiness effect here. Okay, so all of this is working together to make our platelet plug bigger so that we close over this hole. Now, here's what would be happening at the neighboring sites. So even right next to our site of injury, if we step over, we want to make sure that we don't have we don't have our platelet plug expanding and expanding and expanding, clogging everything up, potentially even clogging all the way across, right? So here's where we're producing that prostacyclin and nitric oxide from the healthy cells so that we're constraining that platelet plug right here in the center. We're preventing platelet aggregation at those sites where we don't have an injury. That is really the quick and dirty of our platelet plug formation. Questions about this process right now? Okay. We'll come back to this in a little bit at the end when we talk about some disease states. But for right now, we just want to be able to identify some of the molecules that are associated with this platelet plug formation, knowing that we have platelets adhere and then platelets aggregating. Take a look. Okay. So the correct answer is nitric oxide, because the nitric oxide is about the prevention of the aggregation. So it's coming from healthy endothelial cells. So it's preventing this process of platelet plug formation. The von Willebrand factor is associated with this because it's kind of kicking the process off when it bound to the collagen initially and allowed those platelets to do that first step of adhering to the site of injury. So it wasn't in the picture, but it is necessary for this process to occur. Okay. That's a platelet plug. it's not sufficient to totally heal you. So we need to form a blood clot next. So when we talk about clotting, and a synonym really is coagulation. When we talk about coagulation of blood, we're talking about how it clots and forms a kind of solid gel-like structure, mainly made up, or at least partially made up by a mesh network made of something called fibrin. So this occurs around the platelet plug. So it's occurring at that same site of injury where we've started off with the platelet plug. And the platelets actually do give off some signaling molecules that tell your body, we want to clot here in this place. The clot is really the dominant part of your hemostatic defense mechanism, aka it's the main thing stopping the blood from getting out. It's the main thing that's keeping your blood in the body, although these earlier parts, the vasospasm and the platelet plug formation, help a little bit, help kind of staunch the blood loss. Okay. So when we talk about formation of a blood clot, we're going to see big flow charts of lots of coagulation factors getting activated by other coagulation factors. And I want to make sure that we're paying attention to the most important parts first. Okay. So got that most important part right at the bottom here, right? So what we're going to see as we are forming our blood clot, our stable blood clot is made up of fibrin in a stabilized mesh, right? So we're going for fibrin, right, in a mesh. We can think of that as stable, right? Okay. Now, to get that fibrin stable mesh, we're going to need to make some fibrin in the first place. So fibrin is going to start out a little looser. Okay. All right, that's our fibrin loose. Okay. And to get that loose fibrin, we're going to need to be activating a precursor molecule. Okay. So fibrinogen, really just rewriting what I wrote on the slide here, right? But fibrinogen is kind of an inactive form of fibrin. Usually when you see this inogen ending on a word, that kind of tells you it's an inactivated kind of precursor molecule to something that later on is going to have an active state. So fibrinogen is like our inactive fibrin. So we're going to first have to convert it into our basic sort of active form of fibrin that's not stabilized yet. And ultimately, we're going to have to turn it into a stable meshwork. Right, so that is our big goal of all the coagulation factors and all the processes we're about to look at, is just to make this stable fibrin mesh from initially our inactive fibrinogen. So when you are studying these clotting factors, if you get overwhelmed, go back to that. This is just a picture of kind of what's going on overall. So we have these variety of clotting factors all swimming around in your bloodstream all the time. They're coming from your liver. Here we can see that stabilized meshwork in a clot. It happens to also like catch red blood cells and trap them in there to form part of the body of the clot. Okay. We got lots of pathways. Our clotting factors usually have numbers on them. Very unfortunately, they numbered them, I think, in order that they discovered them rather than the order in which they act in your body. So unfortunately, they're not going to go 1, 2, 3, 4, 5. Okay. So we are zooming in. We're paying most attention first to this bottom line, right? So that's what we just said, that we have inactive fibrinogen that we first turn into a loose mesh, and then we have a stabilized mesh to have our thrombus, to have our clot, okay? So stepping back to figure out how we do all that stuff, we're gonna need to think about how are we activating the fibrinogen in the first place? And then how are we stabilizing it? Okay. So if we step backwards, you can see all of the molecules on these diagrams that are circled in kind of like a pale pinkish peachy color, those are inactive forms of clotting factors. So your liver is making clotting factors. They're always in your blood. You don't want to be clotting all the time. So when they're swimming around in your blood normally, they're in an inactive state. So fibrinogen is one of those. So in order to take that inactive form, fibrinogen, and activate it into this first loose form of fibrin, we've got a molecule called thrombin. All right. So thrombin is directly converting fibrinogen into fibrin. Okay. We need thrombin to directly convert fibrinogen into fibrin. Cool. We're going to ignore this upper part for right now. And we're going to think about that second question mark. So if we converted fibrinogen to fibrin using thrombin, how are we stabilizing that loose fibrin into a stable mesh? The answer is, it's also kind of thrombin doing this, but it has a slightly more indirect pathway. So thrombin isn't going to directly act on that loose fibrin to stabilize it. What it's going to do, we got a little domino flow here. We are activating another clotting factor. So whenever you see these numbers, that's some clotting factor. This one happens to be clotting factor 13. Thrombin activates it. And then that clotting factor 13A for active is stabilizing the meshwork. Okay. Cool. So thrombin has important jobs. So our main clot is made of fibrin, which originally was fibrinogen. Thrombin directly converted that into our active fibrin, and it indirectly gave us a stabilized meshwork with this intermediate clotting factor kind of helping along this factor 13. Okay. So now that we understand the role of thrombin, we have to think about where thrombin came from. Okay. So thrombin also has a precursor molecule in an inactive form. So prothrombin, another way you can tell something is going to turn into something else is that pro prefix usually means we've got a precursor kind of molecule here. Okay. So thrombin needs to be made in your bloodstream from prothrombin. Okay. So that's swimming around as well. Okay. And the way we turn prothrombin into thrombin is through factor 10. Okay. So that is another clotting factor. Okay. So as you'll notice, some of these molecules have been given like names that are spelled out in letters, and some of them are just numbered. So we are going to kind of prioritize which of these factors we want to focus on. So you definitely want to know all these words, the fibrinogen, the loose fibrin, the stable fibrin. You definitely want to know prothrombin and thrombin. Of the numbered clotting factors, the one I would consider most important for you to know is this factor 10, this X here. Okay. And I know it seems like I circled almost everything on this diagram. I did, but there are going to be a lot more numbered factors when we look at the intrinsic pathway and the extrinsic pathway. I'm not going to circle very many things when we add those up. So you want to know about factor 10. You want to know about the relationship between prothrombin and thrombin. You want to know thrombin is directly converting fibrinogen to fibrin and indirectly stabilizing that fibrin through the activation of another clotting factor. So we can see also on this diagram that we've got a positive feedback loop here as well. Clotting is all about positive feedback because we want to close over sites of injury as fast as possible. So once we have our thrombin activated, you can see it's looping back up and circling around and around and helping out with its own activation earlier. Okay. So this is the central part of forming a blood clot, right? So our stabilized fibrin meshwork is a blood clot. Okay. These are the molecules that are most closely associated with the production of that clot. But we do have whole enormous domino effects that ultimately get us to this part. So you'll notice at the top, we have an intrinsic pathway and an extrinsic pathway leading into this final formation of the clot. So we will talk about those as well. Okay, but I want us to note that we're all coming together there to that second part. Okay. So remembering, how did we get here in the first place? Okay, so before we think more about these clotting factors, let's just remind ourselves what are the series of steps we've talked about so far today to get us to where we are so far, which I won't say out loud because I'll kind of give it away. Okay, let's take a look. Okay, good. Yeah, so far today, we've had our platelets adhere to the site of injury. So they stuck there in the first place. They secreted a bunch of signaling molecules that led them to aggregate, to group together into the platelet plug. And now we are finally forming our actual blood clot, our fibrin clot. And it's that fibrin clot formation we're kind of focusing on right now. Okay. So what we've talked about in terms of that fibrin clot formation so far, right, our big goal is here down at the bottom. You see why I've been stepping you backwards. Okay. Fibrinogen into loose fibrin into stable fibrin. And we talked about all this bottom part as well, right? So we now know that we need to turn prothrombin into thrombin so that the thrombin can convert fibrinogen into fibrin and indirectly stabilize the fibrin. And we know that factor 10 is the one that's doing that conversion of prothrombin to thrombin. Okay. So basically, you can kind of think of factor 10 as almost making our thrombin from prothrombin. So our intrinsic and extrinsic pathways are both going to lead us here. So we're going to have an intrinsic pathway that's kind of coming from the blood vessel itself. So like we talked about damage to the blood vessel leading to our platelet plug formation, we're going to have damage to the blood vessel leading to this intrinsic pathway. And we're going to have an extrinsic pathway that's coming from the tissue surrounding our blood vessel. Okay. We're looking at that intrinsic pathway first. Okay. So note for both of these pathways, we're coming in at the bottom to that point where we get our factor 10 activated so that it can make us some thrombin so that we can make fibrin and then stabilize fibrin. Okay. So when we're thinking about the intrinsic pathway up here, yeah, our intrinsic pathway, the trigger for this intrinsic pathway is contact with collagen, right? So just like with our platelet adhering and then aggregating, we have our blood vessel damage exposing collagen. We know that we had von Willebrand factor touching it and making it sticky for platelets. That whole process is also activating this factor 12 up here. Factor 12 activates factor 11, activates factor 9. Factor 9 works together with factor 8, some calcium, and a platelet factor. So that PF is platelet factor 3 to activate factor 10. Right. And this is what I mean. We're going to only focus for our purposes on a couple of these factors to make it more manageable. So we're focusing on the fact that what we're trying to do is get factor 10 so that we can make our active thrombin, so that it's converting fibrinogen to fibrin and then helping us stabilize that meshwork. So that's what's happening with the intrinsic pathway. And we've also got an extrinsic pathway. So what's going on with the extrinsic pathway is that surrounding your blood vessel that's damaged, you have other stuff, right? So that blood vessel is running through something. Right, so we've got tissue surrounding it. So we've got some kind of tissue over here. All right, we've got tissue over here. All right, in order to get a cut, right, if you think about getting a cut on your arm or something, the first thing that gets damaged is not like a capillary or an arterial or an artery, right? You went through something first, right? If nothing else, you went through the skin, you went through the epithelium, if we're talking about external damage. Okay, so that means that we also have damage to surrounding tissue here, right? So I've got damage here going through the tissue. Okay, so when we have damage to that tissue, that's what's creating this extrinsic pathway to clot formation. So the intrinsic pathway is all about the blood vessel itself, things in the blood vessel in the bloodstream. Our extrinsic pathway is about signals coming from damaged tissue surrounding that blood vessel. It also works through activation of various numbered factors. So here we have our inactive tissue factor 3 and our inactive factor 7 coming together with calcium ions to form a tissue factor complex with this factor 7. But their main job is to activate factor 10, which is going to give us thrombin, which is going to give us fibrin and then stabilize it. Okay? So we're all coming into this kind of central part of our domino flow here. So intrinsic factors coming from actual blood vessel damage, extrinsic factors coming from the surrounding tissue that has also been damaged. All of this coming together to activate factor 10. Factor 10 turning prothrombin, which is inactive, into thrombin that's active. And then thrombin turns fibrinogen into fibrin and indirectly stabilizes that thrombin or that fibrin meshwork. The words we're remembering in general are like these ones. Okay. Questions about this process? Okay, great, because I think I told you what I know about blood clotting. All right, so we're gonna go through a couple kind of extra details, but just remind me so that I know we're all on the same page. What is the role of that intrinsic and extrinsic coagulation pathway here? What was our kind of difference or similarity? Okay, let's take a look. Okay, yeah. They're coming from different places. Intrinsic coming from blood vessel damage, contact with collagen, extrinsic coming from damage to surrounding tissue, but ultimately they're both going to act the same way when we get down to the nitty-gritty of how are we actually forming the clot, because they're both activating factor 10, which means that thrombin can now convert fibrinogen to fibrin and then indirectly allow for the stabilization of that fibrin. Great. Okay. So when we're thinking about all those many clotting factors, those clotting factors, they're coming from the liver. The liver has many jobs. Producing clotting factors is one of them. We saw in our last unit, I guess, or last month, we saw that the liver was also involved in these recycling of our red blood cells. We also think about the liver when we think about kind of filtering out toxins sometimes. Liver does lots of stuff, but one of the super important things it does is let you make blood clots. So these clotting factors are ultimately coming from the liver. They're put into your bloodstream in an active form, waiting around until they get activated in this big domino effect, in this cascade. Now, sometimes we do have plasma without clotting factors, like you might give someone serum. Serum is plasma where we've removed those clotting factors because we are not trying to give someone a blood clot or increase their risk of clotting. Okay, so we can medically remove those clotting factors if we're giving like transfusions and stuff. Clotting factors are not the only thing that we need in order to form a blood clot. So we saw on there that we had some little helper ions and some helper factors. So particularly calcium and this platelet factor 3 that is coming from those platelets that have been adhering and aggregating and forming that platelet plug. They're also part of the trigger for this clot formation and part of this activation process. Just like with the platelet plug, we do have factors that limit clot formation because clots can be dangerous, right? People do form clots when they don't want to sometimes, right? Like, and if you form a clot in, say, a vein in your leg when you're sitting around in an airplane, and then it breaks off when you're walking around, it might get stuck in a blood vessel in your heart, give you a heart attack, it might get stuck in a blood vessel in your brain, give you a stroke. We do want to limit clot formation. Right? It's good if you've got an injury. It's bad if you don't. Right? So we have factors for that. So in general, when we talk about anticoagulants, we're talking about things that prevent coagulation, coagulation being the formation of this blood clot. So we have some anticoagulants that are proteins in the plasma and on the surface of endothelial cells. So just like those healthy endothelial cells, we're preventing the aggregation of platelets. We also have anticoagulants preventing or limiting clot formation. So one way that we can inhibit clot formation is by inhibiting that extrinsic pathway, that signal coming from the tissue. So damaged tissue has a bunch of tissue factors, ultimately activating factor 10 and leading to our whole clot formation, that formation of the fibrin clot. But if the tissue is healthy, it should be secreting tissue factor pathway inhibitor from some healthy endothelial cells in that tissue, kind of stopping this extrinsic pathway and therefore limiting how much active factor 10 you have, therefore how much fibrin you have, thrombin you have, and how much fibrin you have. Another anticoagulant, another molecule that is preventing too much clot formation is something called thrombomodulin. So thrombomodulin is also coming from healthy cells, from healthy endothelial cells. And what it does, if you have these healthy cells secreting lots of thrombomodulin, they like modulate the activity of thrombin, right? So modulin and thrombo. Okay. By that, I mean that the thrombomodulin, when you have these healthy cells secreting it, binds onto thrombin, and then thrombin's not going to do its job of converting fibrinogen to fibrin and then stabilizing. Instead, that thrombomodulin binding to thrombin is going to form a complex activating another protein called protein C, which is an additional anticoagulant. So in both platelet plug formation and in the formation of a true blood clot made of stabilized fibrin meshwork. We have healthy tissue and healthy cells in the lining of our blood vessels, secreting signaling molecules that stop us from activating these various clotting factors and forming a large clot. Now, once we make a blood clot, we do also want to break it down. You don't want there to be a blood clot at a site of injury forever, right? You wouldn't like having a scab just on your arm forever every time you get a cut. So we are also going to dissolve our blood clots. Perhaps not surprisingly, we have an inactive molecule here called plasminogen that needs to get activated, gets activated into something called plasmin, and then plasmin breaks down fibrin, right? So to make the clot, we had fibrinogen, to our loose fibrin, to our mesh fibrin that's stable, big clot. Okay. When we're dissolving it, we are going to have plasminogen. Here's why I was giving you that tip on that inogen ending, right? We'll see this come back when we do GI stuff as well. So plasminogen turns into plasmin. And that plasmin is going to go ahead and break down fibrin and attack our clot, right? That's the job of the plasmin. Now, one of the ways that we convert plasminogen to plasmin is through our tissue plasminogen activator. And that's actually secreted by the endothelial cells lining your blood vessel while they are forming the clot, right? So while we're making this fibrin meshwork, the endothelial cells are also giving off some of these plasminogen activators. So we can kind of think of that fibrin meshwork as activating our plasminogen, right? Activate, creating plasmin that then ultimately breaks it back down again. So it's basically a negative feedback loop here. We're activating this molecule using fibrin that is ultimately going to destroy our fibrin. Okay. So that's the big picture process, right? So overall, we have a vasospasm happen first. Then we think about platelets. So we activate our von Willebrand factor, which allows platelets to adhere. Platelets aggregate. They give off various signaling molecules, causing that platelet plug to expand and get bigger. So it even tightens a little bit. As they're doing that, they're giving off some platelet factors too that are helping along the activation of this blood clot formation, this coagulation pathway, which ultimately boils down to the fact that we need to convert fibrinogen into fibrin and stabilize it. Okay. So we do have various disorders that prevent blood clot formation in a couple different ways. So two that work kind of similarly are hemophilia and von Willebrand's disease. Okay, so in both of these, we're going to end up not forming clots appropriately because of genetic disorders that cause some kind of deficiency. So in hemophilia, that's a genetic disorder that is caused by deficiency in genes creating one of these coagulation factors. And it's usually, I just pulled back this picture here. It's usually, I think, this factor 8 here. You don't need to know that specifically, but what's happening is that when we don't produce working form of any of these clotting factors or coagulation factors, right? If we stop this pathway, ultimately leading us to our stable fibrin meshwork, then we're not going to form a stable fibrin meshwork, we're not going to have a blood clot, and you're at risk of bleeding out. Okay. So in hemophilia, it's for a specific coagulation factor involved in Willebrand's disease. The problem is with our VWF, our von Willebrand factor, which was associated with that platelet plug formation that happened a little earlier on. Von Willebrand factor also does partially play a role in kind of carrying around some of these coagulation factors, specifically, I think also that factor 8. So you can see that there's also a platelet factor here, kind of this step as we're activating factor 10, right? But ultimately, if you decrease platelet plug formation, you are also kind of ruining your pathway leading to a stable blood clot. Okay. So we can do it through directly having a malformed coagulation factor, or we can do it through not forming our platelet plug appropriately. The third thing on this list is kind of more dietary things. So you do need to synthesize these clotting factors. You need to make them, right? Your liver is making them. And it needs certain materials for that process. So in particular, vitamin K deficiencies can lead to lower levels of clotting factors because you don't have the necessary components to make those clotting factors. So all of these would lead to higher risks when you get injured. Finally, when we think about blood clots, often we think about blood clots as being dangerous because they lead to risks, in particular of heart attacks when we get clots breaking off and blocking one of those coronary arteries that you'll be looking at in lab. And a common kind of treatment for this can be giving like baby aspirin if someone has had a heart attack or maybe a stroke for its anticoagulant properties. Okay. So what's going on with that is that this is a dose-dependent effect. It's not a more is better kind of effect. So at low doses, aspirin is going to act as an anticoagulant because it kind of downregulates our platelet aggregation. So what it does at low doses is it stops us from forming that thromboxane A2, which was one of our kind of like helper molecules that our platelets are secreting as they're aggregating and doing their positive feedback and making a bigger and bigger platelet plug. Okay. So at low doses, if we don't make thromboxane A2, we're not going to make quite as much ADP. So those platelets aren't going to aggregate so much. So that platelet plug isn't going to get so big. Maybe that platelet plug isn't really going to form. Okay. So that's how it works as an anticoagulant, but it's only if it's at a low dose. If you give someone a higher dose of aspirin, it's going to have another effect. So higher doses of aspirin are going to affect prostacyclin. Okay. So if you take too much aspirin, you're going to inhibit the formation of prostacyclin, which was one of those two molecules that your healthy endothelial cells are making, the undamaged ones. That was one of the things that was stopping platelet aggregation. So if you give high doses of aspirin, you might actually be making more clots. So if someone tells you to take a baby aspirin, take a baby aspirin, not like 5 big aspirin. Okay. So we're going to stop there and we'll talk about anatomy of the respiratory system next time. So make sure you're familiar with our conducting zone, our respiratory zone, and in general, the beginning of that chapter.